afternoon, ladies. Today we're going to talk about cell transport, and it is not the world's easiest topic, so if you have any questions throughout the video, please jot them down, and we can discuss that when we meet again. The biggest thing I want you to understand is vocabulary here is really important. Making sure that you understand what each word means and how it relates to how cells are transported is going to be critical in your understanding of transport. We're going to do a couple of activities that will hopefully make it easier in class, but please make sure that you're understanding all of the vocabulary that we talked about, or that we will talk about. Also, there are going to be 2 activities I'd like you to do. Please do those in OneNote in your homework tab. That way I can keep track of what you're doing. We'll need one of the activities for class on Thursday. Our learning objectives are to explain how passive transport and active transport work when you're talking about cells. So please make sure that you understand that cell transport is defined as how things enter and leave the cell. Passive transport, if you look at the word passive, means that there's no energy being used, whereas active transport is going to require energy. When we talk about the cell membrane, remember back to the fluid mosaic that we discussed and how there are channels and pumps. These channels and pumps are important because this is one way that the material moves across a membrane. We'll talk about that a little bit more kind of as we move on, but permeability is a really important term here. Permeability basically explains what can and cannot pass through a membrane. Speaking specifically to the cell, there are 3 different types of permeability. You have cells that are permeable, which allow crossing of molecules, impermeable, which do not allow the crossing of molecules, or selectively permeable. Selectively permeable only allows certain types of molecules to pass through. We're going to talk about diffusion first. So when we talk about passive transport, remember that I said there's no energy required. So what I want you to do is imagine that I was at the front of the classroom right now, and I was spraying air freshener in the front of the classroom. If you're sitting in the front of the classroom, what do you think is going to happen? Are you going to get an instant puff of the scent, or is it going to take a while to get back to you? And then think about if you're sitting in the back of the classroom. Will the effect of the air freshener be instantaneous, or does it have to, what do you think happens to those particles? How do you think they're able to get back to the very back of the classroom? The tendency for things to be bunched together in a high concentration and then move into a low concentration is essentially diffusion. If you check out this picture right here, you have a cell membrane. On the bottom of the cell membrane, you see a couple of molecules, but then on the top of the membrane, you see more molecules. The top would be deemed high concentration, and the bottom would be deemed a low concentration. When you're talking about different types of solutes and solvents, what I keep saying in my head is solute is minute. So solute is going to be the smaller amount of something that dissolves in a larger solvent. This becomes important when we talk about osmosis specifically. So solute is usually going to be related to the particles that are dissolving inside of a solvent. You can see here that outside the cell we have a higher concentration than we do below and inside the cell. What happens is diffusion causes net motion. The solute particles from the side with higher higher solute concentration will then travel to the side with lower solute concentration. Remember, the body likes to maintain homeostasis. It likes everything being even. So when we're at equilibrium, when we're at homeostasis, particles will move equally in both directions. There's not going to be any kind of a net change. That is one type of diffusion. Then we have another type of diffusion that's called facilitated diffusion. The facilitated diffusion happens more specifically if you look back to the fluid mosaic we talked about. When charged or large molecules try to travel through protein channels, there's no energy that's needed, but because the molecule is larger and charged, they need assistance. So if you think about this cell membrane right here, these solutes are small. There are some solutes, and I can't figure out how to write because my computer's being weird, I'm sorry. So if you imagine larger circles right here, larger different types of solutes, they can't fit through this membrane right here. So you have these built-in channels, and the channels allow charged and larger molecules to pass through. You still have a change occurring due to the concentration, but it's important to know that there is still no energy that's being used when you talk about facilitated diffusion across a cell membrane. When we're talking about osmosis, I'm going to have you guys watch a Khan Academy video that will explain osmosis a little bit better in a way that you can understand really well. I thought it was really interesting the way they used all of the pictures. And I want you to pay attention to how they're presenting, because this will play into what we're going to do on Thursday. But basically, osmosis is diffusion of water. It happens in the aquaporin, which is where the water diffuses. And what is happening here, you'll get more information in the osmosis video, but what's happening here is you can see that the sugar, which is the solute, remember solute is minute, solute is the smaller thing. If you look, you have a higher concentration of sugar on the right side as compared to the concentration of sugar that's on the left side. What happens, because you have a selectively permeable membrane that does not let sugar pass through, water passes through. Again, the body really likes to maintain normalcy. So water passes through on one side, but you still want an even concentration. So you can see the water levels end up being different because you need more water to be able to have the same concentration where there is more solute. You need less water to maintain concentration when there is less solute. So you can see instead of the solute switching through the permeable membrane, you have the water switching through permeable membrane. Osmosis really depends on isotonic, hypertonic, and hypotonic concentrations. What I want you to do is really look in the textbook to get a good understanding of the illustration here. Make sure you're reading everything. But we have isotonic, and if you think of iso, that's the same, right? So isotonic has the same concentration inside and outside of the cell. So water can freely move inside and outside of the cell. You see here an image of an animal cell and an image of a plant cell. Water freely moves inside and outside of that cell because there is no concentration difference. When you're dealing with something hyper, you're going to end up with something, hyper means above. So when you have a hypertonic solution, the higher solute concentration is in the solution as opposed to in the cell. So water has to leave. Water has to leave the cell to help balance out the higher concentration that's being found outside of that cell. And then if you think of hypotonic, hypotonic means low, so inside of the cell there's higher concentration, whereas outside of the cell there's lower concentration. So you're going to end up with water coming in to help make sure that everything is the same. What I want you guys to do in your OneNote is basically, and I'll put an image at the end of this so you understand what I'm looking for. I want basically the same chart. I want isotonic, hypertonic, and hypotonic here. And I want you to draw an image of a cell. So you're going to have just 3 different cells. You don't have to make them specific to plant or animal. 3 different circles, okay? I want you to draw a solute in a solvent with these types of concentrations. So isotonically, you're going to have the same concentration of a solute inside of the cell as outside of the cell. Hypertonic, you're going to have more solute outside of the cell than inside of the cell. And hypotonic, you're going to have more solute inside of the cell than outside of the cell. When I say more solute, I mean the concentration, right? You're going to have a higher concentration for hypotonic. That higher concentration is going to be inside of the cell, okay? And to be able to even this all out, osmosis works with water just as it did in this slide here. The water switches, the molecules don't switch. So another example of this that I'd like to share is if you're thinking about fertilizer. If you're thinking about fertilizer, you if you're thinking about no, we're not going to do that. We're going to just split that out. Nope. The last thing we're going to discuss is going to be active transport. And active transport is where you actually have energy. So remember all of the things we just discussed in terms of diffusion, facilitated diffusion, and osmosis. This is all forms of passive transport. Now we're going to discuss active transport. That's where you have energy that's actually being used, usually in the form of ATP. Molecules that need to move up a concentration gradient, remember the body doesn't like that. It's not easy for the body to do. So you have to have energy being used to really make something facilitate up a concentration gradient. So what if a cell needed to move materials against the gradient from an area of low to high? This is where you would need energy to work against the natural flow of diffusion. Energy is needed to get these substances into or out of the cell because the membrane is selective. For some substances that can't pass through the membrane freely, we end up using this active transport that again requires energy. Protein pumps, which you can see featured right here, is a form of active transport. It uses ATP, which is a type of energy, to pump small molecules and ions across the cell membrane. Active transport proteins change shape during this process, binding substances on one side of the membrane and releasing them on the other. An example of this would be a sodium-potassium pump that has to do with how your muscles work. We then have something called endocytosis. Endocytosis you can see happening here. That's where a vesicle is formed around a molecule that the cell wants to expel. That vesicle is then released fully, including the whole vesicle, out of the cell. And then when we have exocytosis, that's when you have the vesicle that contains the material that you want to expel from the cell, but when it expels from the cell, that vesicle then blends into the cell wall and the vesicle is not expelled. Just the item that you do not want inside of the cell is expelled. So you have, again, endocytosis, which you see featured in the middle, and then exocytosis at the end. And you can really see this illustration here. The whole vesicle is expelled. Whereas here, you're really just talking about the item, the molecule that you're looking to get rid of. So if you think about something that's called phagocytosis, is actually one of my favorite words in biology, which I realize is probably kind of strange that I have a favorite word in biology, but whatever. Endocytosis is really a great example of phagocytosis is really a great example of endocytosis. Phagocytosis is basically if you would think about the white blood cells in your body, this is a great example of phagocytosis. It basically engulfs a cell and then breaks it down. And it's a good example of endocytosis because a membrane pocket forms around the particle that you want to break down. The pocket then breaks loose from the outer portion of the cell wall, forms a vesicle within the cytoplasm. Another really good example of phagocytosis is if you think about amoeba, they use phagocytosis to engulf their food. Why do you think endocytosis is an example of active transport as opposed to facilitated diffusion? The reason here is endocytosis is going to require energy to engulf a particle and form a vesicle around it. After you guys watch this video, I want you to work on the chart, which I will... Ladies, this is an example of what I want you to do in your OneNote in your homework section. You're going to have an isotonic, a hypertonic, and a hypotonic column here. And as you can see, I've drawn a very rough example of a cell. I want you to have one color that is your solute and one color that is your solvent. Look to the Khan Academy video as it kind of explains it very well in terms of kind of imagery that I'm looking for. But in isotonic, remember we have the same concentration of solute inside and outside of the cell. So you're going to draw your solvent around in forms of little molecules, so little circles, little squares, what have you, and then use a different color to represent your solvent. I'm sorry, I said that backwards. For your solvent, draw molecules around, and then your solute, which is the particle, represent that as well in an isotonic manner. So where you have the same concentration inside and outside of the cell. For hypertonic, make the appropriate adjustments, and for hypotonic, make the appropriate adjustments. I want to make sure that you guys put this in your homework folder so that I can look at it at the end of class. The last thing that I want you guys to do is focus on active transport, and I'd like you to explain the concept of exo and endocytosis in picture form. You can do that however you would like, and I want you to voice over and give me a recording of what is exactly happening in your own words. Explain endocytosis and explain exocytosis.